So, around 20 past now. It's not. 10 minutes worth that. Okay, we'll stand here. Okay. Thanks all for coming, and I hope there's a few people who joined online as well. It seems like it. That's nice. For those of you that might ask yourself, who is that guy? I'm a postdoc fellow. I'm Jeppe Christensen. I'm working with Jet Vinder on, and a few other people in the room as well on, well, rewilding projects around the world. And one of the projects has taken me to Montana. And some of you, those of you that are lucky enough to do field work will know that sometimes you meet very great and inspirational people out there on those field stations, and that's actually how I met Ellen this summer in Montana at this fantastic bison rewilding experiment on the Great Plains out there. So Ellen, thank you so much for coming over and giving this this talk. So Ellen has a PhD from Kansas State University, and then you did a postdoc first with the Kaspari lab in Oklahoma. It's quite a renowned lab, both doing grassland ecology, but also actually tropical forest ecology. Similarities with the ecosystems lab here at Oxford. And then you went to Germany to the Senckenberg Institute to do another postdoc, and then finally you got a position at the Smithsonian at their new, what is it called, Grasslands? Great Plains Division
program.
Oh, okay, great. Yeah, as so far the only non-mammalian biologist, or what is it? So you're, yeah. But anyways, it's sort of hopefully something that's building up. But you're part of that process, and yeah, and I really hope that that's going to be a great success, because yeah, there's still a lot to learn on the Great Plains and about grasslands in general, and you have used insects as a lens to try to understand some of the processes going on in those environments, and we look forward to hearing about your research. So thanks.
Thank you. Yeah, and thanks everyone for coming. So yeah, I'm really excited. It's a big honor to be. Hopefully, get to share some of my research with you. I think there'll be plenty of time for questions at the end too. So hopefully, you can be thinking about what might be interesting to talk about next. So I'm going to talk about 3 different projects with you today. The first is about sodium shaping food webs. So sodium is a really understudied nutrient that we don't know that much about, and there hasn't been very many studies on it, but we have some evidence to believe it can play an important role in structuring food webs. Then I'll be talking about this nutrient dilution hypothesis, which I'll get more into, but it's basically this idea that with more carbon dioxide in the atmosphere, plants have this CO2 fertilization where they tend to grow larger faster, and they can suck nutrients out of the soil and actually be depleted in plant quality, which then can in turn affect herbivores as well. And then last, I just started this new position in Montana, as Jeppe mentioned, this last year. Preliminary data from the summer, which is primarily looking at how some of these large mammalian grazers affect both plant and insect communities in this system. So grasslands are a pretty imperiled ecosystem. Depending on how we define them, they can cover between 20 and 40% of the Earth's landmass. And we think only about 5 to 10% are intact. And the main way we measure this is if the grassland has been plowed or not. And even more in a bad situation is tall grass prairies. So these are highly fertile ecosystems with a huge amount of topsoil, and they've been almost entirely converted to row crop agriculture. And so they're considered one of the most endangered ecosystems on the planet. And then when we have grasslands, we have natural grasslands, we still have quite a few big threats. So one of the main threats is suppression of fires, because a lot of grassland urban settings, which again is related to them being converted into cropland. And in these systems, we don't want to have our homes burned down. So we prevent fires, and then the grasslands eventually turn into shrublands, or eventually over longer periods of time into forests. And another major problem in the remaining grasslands is the lack of these large native herbivores. And even when we have those, we don't ever, in almost very few situations do we now see the natural migrations of these large herbivores. So we have them in penned areas, and it's probably a pretty different dynamic than we had in the past. And then, of course, we have some other big problems that are happening regardless of the ecosystem you're in. So we have major changes to our biogeochemical cycles. And of course, most infamously, we've been greatly altering our carbon cycle, which has resulted in climate change and many other effects. It's not just carbon. So this is a framework that's been proposed by Stefan et al. In 2015, the Planetary Boundaries framework, which shows some different ways in which we've changed the globe and predicts that there are some of them we've surpassed. And of these, some of the ways that we've perhaps most passed the boundary of what's sustainable are changes in our phosphorus and our nitrogen flow. So these are kind of we know what we've ever seen before on the planet or have measured. And the only other proposed type of way we've changed the Earth to this degree is this, what's up at the top, which is actually the rate of extinction. But then again, it's not just carbon, nitrogen, and phosphorus. So we have quite a few other elements that we are changing their cycles as well. On our periodic table, we have about 25 elements that are critical for some form of life, and many of these are being altered in pretty drastic ways. And micronutrients in particular might be particularly important for arthropods. And this is a meta-analysis that I was a part of where we looked at fertilization studies, and we specifically in grassland systems, and we were able to get enough data on fertilization effects with of nitrogen, potassium, phosphorus, and a pretty common micronutrient mixture, which is mostly a lot of trace minerals or trace heavy metals. And so what we found is that when we add different combinations of nitrogen and phosphorus, we have this positive effect on total plant biomass. So here I'm showing you the results as an effect size, which is a pretty common metric I'll be showing in this case. It's an experimental system where a positive effect size means a greater increase in plant biomass compared to our control. So, this is not a very surprising result. So we have fertilized plants, they tend to grow more. But what was a really interesting thing that we found within this study was that this was really contrasting to our response of our insects. So when we add these combinations of nitrogen and phosphorus, we tend to have an increase in plant biomass, but kind of a similar effect size. But with insects, we actually have this much greater effect size when we add the combination of all of these elements, so nitrogen, phosphorus, potassium, and the micronutrient mixture as well. So this may show some indication that it's not just these main macronutrients that we generally think about, but actually this mixture of all of them could be really critical for maintaining higher abundances of insects. So now I'll be going into the first project that I'll talk looking at sodium effects, and also specifically looking at this question of if plants do uptake sodium, do they suffer greater herbivory? So probably, if we put salt on the land, it's not good for plants, right? We have this sowing salt phrase you've probably heard. But in contrast, these large herbivores are very attracted to salt. So we'll put out salt for our livestock, put out salt to attract animals to come. And this is a kind of maybe something from the background of your head, but in fact, it's true that sodium has this interesting dynamic where it is not an essential element for the majority of plants, but it is critical for animals. In fact, it's found in every single cell and every single animal in our sodium potassium pumps. So herbivores specifically have this potential to be highly sodium limited, generally not sodium limited because they eat meat, or even just eating some dairy products or eggs gives you plenty of sodium that you can survive and thrive. So it's fairly unique, probably the only other element like this of all of our elements that is limiting to animals but not plants is iodine. So this might just be these two. And quite different situation than most of the time when we're talking about nutrient limitation, which we generally think of from a plant perspective. And look at those elements that are limiting at that level. And also the sodium cycle has been greatly altered by human activity. Primarily humans are adding sodium. There's greater sodium availability on land than there used to be, and this is occurring in a variety of ways. Probably the biggest one is through saline irrigation. About 20% of irrigated land has been salt, and about the size of France. So really a big problem globally and probably predicted to get worse in the future. And then, of course, road salt application. So in freezing climates, we add quite a bit of road salt, kind of incredible amounts. And this is also predicted to continue as we continue to build roads. And then another one that you might not have thought of about as much is with increases in sea level rise. We also have greater inputs from sodium aerosols. And then just kind of a funny one I wanted to add is that these goats within a national park in the United States, they became very attracted to the hiking trails. There was a specific source of sodium there that they became addicted to and actually became a danger to hikers, and the whole like goat reintroduction project failed, and because they became so aggressive and had to be removed from the park. So yeah, many ways in which humans are adding sodium to the earth. And so sodium's not just used by our large herbivores, it's also important for smaller animals as well. So this was a sodium pulse experiment that I conducted across 54 different grassland sites, where we put out a treatment plot with sodium addition compared to a control plot. And again, showing you the results here as an effect size, where a positive effect size in this case means greater attraction to sodium addition compared to the control. And you can see across our different functional groups of insects, we have a positive effect size, meaning they're attracted to that sodium addition. And this is a particularly high effect for herbivores and omnivores, which makes sense based on our understanding of food web dynamics. So where did these insect herbivores get sodium from? Opposed to the ocean, they can get it from these aerosol inputs. But if they're more inland, some important sources actually do include the urine of larger ungulates. Some of them engage in this puddling behavior where they'll seek out sodium from some wet soils. Opportunistic carnivory is actually an important source of sodium. And then there's many additional strange behaviors that insects will engage in to get sodium. So this is an example where this moth is known to frequently rest on birds at night and drink their tears. So kind of gives you some interesting ideas about how other insects can get sodium as well. But so when we conducted this sodium pulse experiment where we looked at are the insects more attracted to a sodium addition plot compared to a control, since it was across this big variety of sites with varying in sodium levels in the background, we also wanted to test, do these sodium gradients. And so we looked at the attraction to sodium both across a gradient of sodium that was in plant tissue and also across sodium in the soil. So we would predict that if that's a source of sodium for the insects, that they'd have a greater attraction where there's low sodium at that site. And we found a fairly weak but significant relationship between our attraction to the sodium addition plots across the gradient of plant sodium. So in our sites with low plant sodium, they tended to have greater attraction, but not in our soil sodium levels, interestingly. So this could indicate that plant sodium is actually an important source of sodium for insects. So plants, again, they don't require it. Most plants, there's a few plants that do, but the majority of plants don't require sodium as an element. But if it does exist in the environment, they will passively uptake it. And this could be something that insects are taking advantage of. So that if the plants do uptake sodium, that they'll suffer greater herbivory rates compared to those that do not. And so to do this, we conducted a second experiment, which was a press experiment where we continuously added sodium to larger plots. We did this across 4 sites that are north and south along the Great Plains. And we also looked at quite a few different measures of both the insects and plant communities. I'll be focusing on the herbivory sampling today. So this is just, I wanted to show you that we do have uptake of sodium in the plant tissue. So here I'm showing the amount of sodium in both the grasses and our forbs, and this is across our 4 sites with our sodium addition plots in yellow and our control plots in greens. And this is these samples month after sodium addition, the first sodium addition. So we can see that we do have sodium uptake. When you add sodium, the plants will incorporate it in their tissue. And I didn't include this slide today, but a lot of people always ask me about effects on plant biomass. We actually, in a bunch of lab studies, it's been shown that a common phenotype for salt stress is for plants to grow less, but we don't really see that. We didn't see that in this experiment, but this was a very low level of added to mimic the effects of ungulate urine, so not the level that is added in a lot of those lab studies. And then we conducted herbivory scoring. This is using a protocol that was developed by the Nutrient Network, which is a big network across grassland sites where they do similar work. And we categorized herbivory of 4 different types: chewing herbivores, the sucking and mining herbivores. We looked at fungal damage, and we did score vertebrate damage, but it was pretty rare, so I'm not going to include any results on that. So here I'm showing you again as an effect size the amount of those herbivory, that herbivory damage across our 4 sites. And sorry, I kind of changed the names of the sites, but these are our 4 different sites, and below it's showing the grass species that was scored. So unfortunately, we didn't have grass species that were common across the 4 sites, but we chose the dominant species within the site because we wanted it to be a species we could easily collect across all of our plots that was naturally occurring. So we do have these positive effect sizes. So we found support for our hypothesis that with sodium addition, we do have more herbivory on these leaves. And this was particularly true for our mining herbivores. We also saw greater fungal damage on the leaves. And this is a little bit less clear exactly what's going on. There's it could be a direct effect that the higher sodium levels. There's actually not that much known about the nutritional requirements of fungi, but I think it could also be that the plants were stressed and then they tended to be more susceptible to fungal damage as well. And then we had some weaker evidence of some increased chewing damage of the herbivores. So in conclusion, we did find some strong evidence that sodium is an important nutrient that does regulate food webs of plants and insects. So in our pulse experiment, we found that sodium addition compared to control plots will lead to increased local abundances of insects, especially our herbivores and omnivores. And when we add sodium to the earth, we also see greater damage on those leaves, meaning that they're suffering more herbivory. So this could have big implications for additions of sodium from all these forms of sodium that the humans are adding to the earth. And yeah, it could alter the food web quite substantially. And yeah, I'm not going to talk about this experiment too much, but I just wanted to give a quick plug. So this was an experiment on how sodium affects grasshopper physiology that was led by an undergraduate in our lab. So she grew grasshoppers in the lab on 3 different diets that were varied in sodium content. And she did this kind of fun experiment where we had them in this grasshopper arena, and we looked at could jump and these things, and we found actually quite interestingly that they grow smaller to a smaller size on a high sodium diet, but they're much more into jumping and even jump further. So kind of, yeah, could be some, we were interested in if they have like greater ability to escape predators, since sodium is quite yeah, sure. Is often used within the muscle function. So now I'm going to move on to the second project, and this is using a long-term grasshopper data set from Conzo Prairie, which is a long-term ecological research site, and looking at how changes in the dynamics of the grasshoppers are affected by both this nutrient dilution and long-term climate cycles. So probably you've seen, if you follow the insect literature at all over the last 5 years or so, there have been this increasing amount of studies on papers on insect decline, and also kind of followed up by also a large number of studies somewhat criticizing these papers on insect decline. So it's an issue that ecologists are arguing about a lot, and there's a lot of challenges in studying changes in long-term insect trends. So why is it so difficult? Well, we do need really high quality data that we often don't have, and getting this long-term data generally less supported by funding cycles. But some other challenges exist as well. For example, insects often have the presence of cycles into their long-term dynamics. And so here's kind of an example of how challenging this can be when we're looking at trends, with the gray line representing our true changes in some measure of insect productivity over time. And then the black line is representing our true trend. And then this red dashed line is what we get if we run our linear regression through the data without knowing much, or just having some of those points along those cycles. And then, so we have this potential of the cycles to cause a bias in our trend. And this could, cause it to look like it's not declining when it is, or look like it is declining when it's not. If we capture less of these cycles over time, we're much less likely to know the true trend, whether that's because we have a shorter time series or because we have longer cycles. And so these cycles are driven by quite a few different factors. So they can be driven by predator and prey dynamics, but they can also be driven a lot of times by these long-term climate cycles like El Nino, Pacific Decadal Oscillation, and the North Atlantic Oscillation. And then, of course, there's another big challenge, which is that there's just so many drivers that can affect these long-term observational data. So here's just a few: global heating, light pollution, invasive species, pesticides, habitat loss, and many more. And these can all be co-occurring, and it's hard to tease out their effects in these observational data sets. So for this study, I wanted to look at 3 different research questions using a long-term grasshopper time series. I wanted to look at if we see these cyclic patterns within the grasshopper abundances over time, look at the overall trend, and then look at the drivers of both the overall trend and the cycles, if they exist. And grasshoppers, a lot of people always ask me, why grasshoppers? What's important about grasshoppers? But they're just a really critical member of grassland ecosystems. They're the dominant herbivore in many grasslands. They help turn over nutrients and decompose plants and break down larger live plants into little tiny dead plants. And they're also this key food source for so many of our members in the higher taxa. The group Orthoptera, which the grasshoppers belong to, has been on Earth for about 300 million years, but became a lot more diverse and abundant in the last 60 million years or so with the rise of grasslands. So they've been in grasslands since the beginning and really shaped this system and are an integral member of this system. And we don't have that much data on many of the taxa in this group. However, in Europe, which is probably the most well-studied area, we know that about a quarter of the species that have had good data are listed as threatened. So there is some indication that we should be worried about this group. So this site was conducted on the Konza Prairie long-term ecological research site, which is located right in the center of the US in Kansas. This site has a lot of really nice long-term data sets that have been collected since the late 70s or early 80s, and that includes long-term grasshopper sampling. And the grasshopper sampling, it has data since the early 80s, although it has a lot more standardized data since the mid-90s. It's a tall grass prairie, and the whole area is an experimental grassland that's been divided up into different treatments of fire frequency. So annually burn sites, 2-year burn, 4-year burn, 20-year burn, and also grazing about areas that are grazed by bison and areas that are ungrazed. And then something that's just so nice to work on in the system is you have these parallel data sets. So we also have a really nice long-term data set in, at least in our ungrazed sites, on above ground plant biomass since the mid 80s. So yeah, just gonna go ahead and jump into the data set. So here I'm breaking down the data set by the 3 different common grasshopper functional guilds. So we generally divide grasshoppers up into those that eat forbs or forb feeders, our grass feeders, and those that eat both are mixed feeders. And text, like diversity-wise, the grasshoppers in the system are pretty split evenly by these 3 groups. And then further within the plots, I'm breaking up the data again into grasshoppers that are in our ungrazed sites and those in grazed sites. So the bison grazed sites are in brown, and you can see that we do tend to have higher diversity of grasshoppers for our forb feeders and also our mixed feeders in the bison area. And this is because bison are predominantly grass feeders. So they eat out the grass, which allows for forbs to grow. And we then have forbs that support these grasshoppers that also eat forbs. And then I would also point out that across these different time series, we actually have a fairly similar pattern over time, and perhaps we can see some evidence of a cyclic pattern and maybe even a bit of a downward trend in the data. So the first question I wanted to address in this study was if we have the presence of these cycles within the data set. And to answer that question, I used a type of spectral analysis. It's called the multi-taper method. So spectral analysis was originally developed. And we have a very robust pattern in this data set for a 5-year cycle. So a cycle with a frequency of 0.2, which corresponds to this 5-year cycle. And it's just a very extremely robust pattern. We can actually find this if we analyze the data in a variety of ways, using different locations at the prairie or even within individual taxa. We can still often get this 5-year cycle. So it seems to be really something that's going in this time frame. So we did check if this cycle is driven by these long-term climate oscillations. The other major way that cycles are often driven is these predator-prey interactions, but unfortunately we don't have as good of data to address that question. So we looked at different seasonal measures, which is the most common approach of the North Atlantic oscillation, Pacific decadal oscillation, and El Nino cycles. And we can get these pretty good correlations between our grasshopper seasonal measures of these long-term climate oscillations. I would definitely say this is correlational, but it is supported also by other research on grasshoppers that they can be driven by these longer-term cycles. And then we broke down the time series. So we tried to get rid of this effect of the bias of these cycles over time. And we pulled out those cycles and looked at our remainder, and were able to calculate a long-term trend over time. And we did find that the grasshoppers in the system are declining by 2% or even 2.7% per year, which is pretty significant and corresponds to more than 30% decline over our sampling period. And so that was actually kind of surprising for me. I was not expecting that in this specific system, which is this well-established research site where we don't have much pressures, decline. So the question is why? Why are the grasshoppers declining? And we don't have habitat loss. We don't have any invasive grasshopper species, and invasive plants are not a big problem. We don't have spraying. Light pollution also is probably not very of a big problem in this region. So what else is going on? Of course, we do have rising temperatures, and this could be contributing to grasshopper declines. We don't have much of a pattern in annual precipitation, so that's what this graph is on the bottom, but of course changes interannually and timing of precipitation could be affecting the grasshopper populations. And then we're really interested in the effects of plant quality, because grasshoppers are just very well known to be strongly driven by changes in plant quality, and we wanted to look at if there were long-term changes in plant quality as well. So why might plant quality be due to changes in climate? And it could also be due to additional carbon dioxide in the atmosphere. So this is the result of a meta-analysis where they looked at CO2 fertilization experiments on 130 plant species. And they showed that with additional carbon dioxide, plants tend to have increased amounts of carbon, but decreases in amount of all these other micro and macronutrients that are critical for many forms of life. And this has also been shown observationally. So this is from a study that's looking at herbarium specimens in pollen, and they plotted the amount of protein in the pollen over the amount of carbon dioxide that was in the atmosphere at the time of that the herbarium specimen was collected. So you can see this is a pretty sharp, strong negative relationship between the amount of protein available and the amount of carbon dioxide in the atmosphere. And the authors of this, potentially this could be affecting bee populations. And then in Germany, I always get this question, like I always get this people saying this doesn't happen in Europe. So I also included this additional study here where there's been a big study across Europe showing that over the last 20 years or so, there is declines in nutrient concentrations observationally in many forests as well. So is this happening at Kansa Prairie? That's the next question. So if we have more CO2 in the atmosphere, what we would actually expect is that the plants are growing, the above ground biomass of plants is increasing, and then they're depleting some of the soil nutrients. We actually see this very strong increase in plant biomass at Kansa Prairie from this parallel data set on plant biomass over time. So we don't have data, unfortunately, from this data set on our graze communities, but we do have it from 3 different sites that have these different fire frequencies. And we can see that fire frequency doesn't have a big effect on this. For the most part, we just have this steady increase in plant biomass over time over the past 30 years or so. So our next step was to go into this old shed and find all these samples that the researchers had been collecting and get some subsamples from all of them, look at the actual nutritional content in those samples. And we targeted some of our big macronutrients that we know are critical for grasshoppers: nitrogen, phosphorus, potassium, magnesium, and sodium. And we also looked at the grasses because we know that these tend to be the same species and will probably be more standardized over time. And we also just did this from our one site, which was our 1-year burn. So we actually did find these pretty big declines in nutrient content in our plants over time, especially for nitrogen, potassium, and phosphorus. We have kind of an odd pattern of changes in sodium over time, but overall does end up as a decline. And then not much of a pattern with magnesium, potentially an increase. And the pattern of magnesium is highly correlated with dry deposition of aerosols. To simplify this analysis, we conducted a quick principal component analysis where we put our elements into a principal component analysis. We got one significant axis, which was then negatively correlated with all of those nutrients that are declining. So our potassium, phosphorus, nitrogen, and sodium. And so we call this axis our index of nutrient dilution. So when we have an increase in this proxy, it means a decline in tissue. Here we're showing this proxy, this index of nutrient dilution as our dashed line over time. So we can see the dashed line is increasing. We have more nutrient dilution over time. And then also comparing it with 2 different measures of the grass biomass, both from the individual site where we collected the data and then overall across the whole site. So we can see that as the grass biomass tends to increase, we also tend to see this quality. And you can also plot this as a regression and see this correlation. And here is again the same measure of our nutrient dilution index plotted with our grasshopper abundance. And we can see that as we have more nutrient dilution, the plant tissue is becoming less nutritious. We also see a decline in our grasshopper populations. So again, observational study, and we need more work on this topic. But based on what we know from grasshopper biology, this makes a lot of sense in terms of nutrients being a key driver of grasshopper populations and could be a cause of grasshopper declines in the system. So is this a larger driver of insect declines? Definitely, we don't know, and we need a lot more work on this topic. However, there are a few indicators that could suggest this could be the case. So there have been quite a few studies that have shown that we don't just have insect declines in our areas that have had a lot of human impacts. We actually see quite a few declines even in natural areas. That's a pretty common finding across a few of our big meta studies. We also might expect, if this were the case, that we would specifically see larger declines in herbivores because they're at this level that faces this issue, and also in grasslands because they're this annual cycle more quickly to the changes in our atmosphere right away compared to forests, which probably have much longer lag time in their response. And we actually have seen that most of the studies, I mean, it's also true that that's the text that most people are working on, but most of the studies that have shown quite a few declines in insects are in the Lepidoptera, which of course are herbivores as caterpillars. And there's also some indication that some grassland systems maybe have some other systems. So this is the last topic I'll be talking about today. So I just wanted, I don't have kind of final worked up data, but especially since Yeppe's here and maybe collaborate with more people in the future on my current work that's now based in the shortgrass prairie in eastern Montana, I wanted to share with you some of my preliminary data of what I've been working on out there. And this is not is based at an NGO site that is called American Prairie. And there's a map of some of the properties they currently own, but it's a big rewilding project and it's expanding all the time. So it's an exciting place to work because there's always a lot going on. There's going to be bison being added to some of those properties that don't have bison yet. And so we can do a lot of exciting before and after studies of and I'm primarily focusing at this site on 4 taxa, which are wolf spiders, dung beetles, grasshoppers, and plants. And the big question from this summer that I wanted to address was, how do these large vertebrate grazers affect our plant and insect communities? So there could be quite a lot of ways that they could affect, could potentially be competing for biomass with our insect herbivores. They also, these large vertebrate herbivores tend to move nutrients around the landscape in quite a few different interesting ways. They provide additional resources for our insect groups, particularly dung and carrion. They also alter our plant and soil structure. And this is really an important topic to work on because our mammalian communities over time as well. So it's another major way that we've shaped the earth that in turn might affect our plant and insect communities. So this summer, I had 2 really great interns, and we did quite a bit of sampling over the season at 15 different sites, which corresponded to 5 different treatments. So 3 reps per treatment of the sites. The treatments of the sites were cattle grazed, bison grazed, ungrazed, and then 2 types of prairie dog. And the prairie dog also was in the bison area, but one was untreated and the second was treated with insecticide. So this is actually a management technique that's being used by the American Prairie because they want to control flea outbreaks in prairie dogs, which then can give the prairie dogs plague and actually wipe them out. And in particular, they're really interested in doing black-footed ferret reintroduction. So if all the prairie dogs die off, we can't really have black-footed ferret as well. So they're currently doing quite a bit of insecticide, which, you know, for me is kind of controversial, but it's also an interesting system to work in because we have the ability to look at how insects shape the landscape because we don't have too many insects in those areas. We did a variety of sampling methods. We used this pasture meter as a proxy of total above ground plant biomass. We did traditional clipping. We also collected insects using sweep netting and pitfall traps. For the dung beetles, we used baited pitfall traps with pig dung, which I don't recommend if you have a squeamish nose, but we did catch a lot of dung beetles. We use ring counts, which is a way to get at grasshopper density. So sweep nets are often criticized because they the sampling in the same way, but they don't give you a true density of the area. So this is a way to get grasshoppers per meter squared. And we targeted plant biomass, chemistry of plants and soils, and maybe Yepe will also help us with some of our additional soil work and add to our study. We did counts of dung to get both changes in decomposition and also the use of the mammals of the site. And then we also in our communities and densities of grasshopper dung beetles and wolf spiders. So yeah, as I mentioned, a lot of this data is still being processed, but I'll give you kind of a rough cut of some of the early things that I found out. And this is a view from my RV. My cat was fantasizing about his breakfast out on the prairie. So here are our changes in plant biomass across the growing season from June to September, and then broken down by our different treatments of ungrazed bison, cattle, and then the red triangle is the treated prairie dogs, that's our insecticide sites, and our untreated prairie dog sites. So pretty expected, as we don't have large grazers, we have higher levels of plant biomass, but also quite interestingly, we have quite a bit more plant biomass in our bison graze sites. And then the lowest levels of plant biomass are found in those prairie dog sites. So the prairie dogs really hammer the system and eat out a lot of the plant biomass. They're both eating a lot, and they of course are dense and abundant in their colonies, but they also will clip out some of the vegetation as an anti-predator technique so that they can see if the coyotes are coming. And then across most of our sites, we also see a peak in biomass in July. So this is a fairly short growing season that peaks kind of early. But I also thought it was super interesting that we don't see this in this treated prairie dogs where we've removed the insects. So this could actually indicate that insects are driving this dynamic of the peak biomass in the middle of the summer. And here are our changes in nitrogen content in both our grasses and forbs and across our grazing treatments. So for our grasses, grass does exist because the prairie dogs do eat it out quite a bit. In our prairie dog sites, we have much higher grass nitrogen. So this is probably a signal of the prairie dogs doing a lot of nitrogen cycling, and also, yeah, mostly through their pooping everywhere, like adding a lot of nitrogen to the system. Our forbs is a bit more of a messy story and is probably a lot more related to which species exist in that system because they're being removed. So grasses is just a few dominant species that are common, but forbs is quite a big diversity of species. We have potentially the highest nitrogen in our forbs in our treated prairie dog sites. So this could indicate that with that spraying, we're actually removing the insects that are targeting those species of high nitrogen forbs. And we maybe have the lowest forb nitrogen in our cattle sites. And this could also be those high nitrogen forbs. And again, cattle tend to eat quite a few more forbs than bison do. So it's kind of interesting result. And here is our overall grasshopper density. So this is from those ring counts. And so we can see that for our treated prairie dogs at the bottom in red, if you spray pesticides, you kill insects. So yeah, that's kind of obvious, but kind of validates this idea that this could be an interesting site to work on where we don't have that many insects, and helps us to look at the effects of what insects do. And then we actually have the most insects in our untreated prairie dogs that are really low plant biomass, but they have this high nitrogen. So this potentially suggests that the idea that the grasshoppers and the large grazers are competing for biomass is not really likely supported. And it's much more likely that there's feedbacks between the two and also it's from a grasshopper ecologist perspective, not very surprising that grasshoppers hone in on high nitrogen, but not necessarily high biomass areas. So looking forward, a lot of work still to do, especially in terms of going through all the samples of the arthropods and getting all the composition data. And this will be a big focus of my winter and spring. And yeah, really interested in these sites that have been treated with insecticide, and they actually just switched to a new form of insecticide there, which is, I'm showing here in the picture, it's these little pink grains, if you can see them, which is fibrinol grain that is the same type of insecticide that you might give to your cat or dog that it's called frontline. And so the idea is that the prairie dogs will eat that, will then be poisonous to the fleas that might bite them. But it's actually also massively consumed by grasshoppers and beetles and ants and everything that's out there. And they all also were wiped out by this treatment. So, and it has a very long half-life, about 4 months. So I think it actually might lead to even greater levels of insect removal. And looking forward, I'm also really excited to have a deeper understanding herbivory on this system, and probably going to be trying to set up some big exclosures in the site so we can do some more direct comparisons in the same soil types. And Yeppe is aware of the challenges of, especially it's very hard to find ungrazed sites in this location. So there's only a few areas that are ungrazed, and probably they have a different soil type. So we want to do some more direct comparison. So overall, broad takeaways from the talk. Of course, we've changed the earth in many ways, especially altering a lot of these large biogeochemical cycles. And this can be a bad thing, both if we have too much nutrients and if we have too little nutrients. So this is really a balancing act where we can mess up things in both directions. And these large mammalian herbivores, which we've also had major impacts on, have many feedbacks with our smaller plant and insect communities, and can be key regulators and even have positive effects on the smaller groups. And overall, arthropods are this key indicator of ecosystem change, but they themselves are also a really critical, important component of the ecosystem that we shouldn't forget, that perform all these important ecosystem services, including decomposition, pollination, and being a key food source for higher vertebrates. So with that, I'd like to thank all the many people who helped with this talk and collect this data. Of course, wouldn't happen without the help of many others. So thank you so much, everyone.
Wow, that's so much to try to take in. But yeah, questions? There's anyone? Mark.
Hi, I have a question about your second study.
Yeah.
You find a higher diversity of arthropods where bison roam.
Yeah.
But you have a higher rate of decline of arthropod diversity where the bison are there. Two questions: is this significant difference in terms of the trend? Was 2.1 to 2.7, I think. Is there a significance there, and what do you think? Is there an explanation?
I mean, yeah, I think it's hard to say. Also, the time series is shorter, so they didn't start collecting the data on the bison area until 16. Well, the time series I had in this study is 16 years. Now it's been a bit longer, so it could be that more recent time period also as a sharper decline. Could look at that as well, but it could be the diversity or the abundance was higher to start with, and that's.
So the two trends are on a per year, are presented on a per year basis, but they are based on different length of time series.
They are, yeah. Okay. So the ungrazed, we have data from a longer time series. Yeah. Yeah. Yeah.
Yep.
Thank you.
Just feel free to just you.
Yeah.
Okay, thank you for the presentation. I have two questions. And when you talk about the, there are some insects that are actually in the hibernation period. Sometimes you may not see them, but all of a sudden you see them placed in a particular site. So they do take note of that to be sure that they were not really hidden somewhere, and you now like say it was a decline. And then for the total information that you get from the research, are you able to translate to the farmers, those people that are keeping the cattle, to be able to know that, okay, this is your result, and this is how it will affect them?
Okay, so yeah, I forgot I'm supposed to repeat the questions as well. So for the people online. So I think the first question is maybe about detection accuracy with the insects. Maybe the decline could be driven by difficulty in detection.
Or, for example, if they have migrated.
Oh, if they're migrating. Yeah. So generally the grasshoppers don't migrate. So I think we can be okay about that. They have an annual life cycle where they tend to stay in the same place. So they'll lay their eggs underground and overwinter as eggs, most species, and then come up in the spring and they stay in the same area so they don't migrate. And then I think this specific data set is quite robust. So there's a lot of sampling in space as well as across time, many subsamples collected. So I think it's, of course, there are biases. Maybe they do have changes in people doing the identification over time, and maybe one person thought it's one species and one person thought it's another. But this specific result is just total abundance of all the grasshoppers. So that probably won't bias the overall abundance data. And does that answer your first question? So then the second question is this applicable to the region and larger ranching community? That's a hard one. So grasshoppers are a pretty hard sell toward the ranching community. They don't, usually ranchers think that this kind of one of the questions I put in here about the competition for biomass. So a lot of ranchers think grasshoppers are bad because they take away the plant biomass and then it's not there for the cattle. They're competing for the forage. So I think, this research shows that we actually have quite low abundance of grasshoppers in ungrazed sites, which suggests that the grasshoppers aren't limited by that total biomass. It's not what's driving their abundances. But in terms of convincing ranchers that grasshoppers are good, I think that's an uphill battle. I'm not sure if I can address that directly with this research, especially, unfortunately. Yeah.
Wouldn't they just say that the reason why there's so many of them is because they've eaten the biomass?
But if they were biomass limited, they would be in the ungrazed. Yeah.
Well, there was other questions as well that you have.
Yeah, it's kind of a basic one, to be honest, but I'm wondering what is sodium used for on sort of a biological level? Why do plants not need it? Animals do.
Yeah, so the question is, what is sodium used for biologically? So the main use in animals is the sodium potassium pump, which keeps our osmotic potential intact in all of our cells. Plants generally, they'll use potassium for keeping that pressure, osmotic pressure, but they don't have a function for sodium in their pumps. There's also within our body, there's some additional function for within our muscle cells and in our neurologically as well. Your brain has like a lot of the sodium in your body goes to your brain. Yeah, this is also like why if you eat a lot of sodium, you'll like swell up. So this is like changing your osmotic potential of your cells. Yeah.
In areas with lots of grasshoppers because of high nitrogen, like higher concentrations of other herbivores, so like bison and prairie dogs in those kinds of areas, do you get general grazing laws for herbivores there, or is it different among grasshopper percentages?
Yeah, so the question is, in areas that have high grasshopper density, because of high nitrogen, do we also see other grazers? I would say we do, although, yeah, it's hard to know what drives that dynamic. So definitely in areas where bison have established for a while, they start to form these grazing lawns, which keep the vegetation low and are nutrient hotspots. And I have a paper on that there's higher grasshopper abundance and diversity in these sites. But that's, I don't know, we framed it as being driven by the bison, not the other way around, but it's definitely possible. It's harder to do an experiment on like keeping the insects out. The main way is to use insecticides. But yeah, there could be more studies on that. That'd be cool.
Okay, I just because I have a very related question to that. Because I also thought about did you know the homeostasis of larger animals, right? Could having large herbivores in the systems, could that somehow moderate that nutrient dilution?
So yeah, just you think they could hear that? The question was, could having large herbivores in the system moderate nutrient dilution? And yeah, I think it could. So we definitely have greater turnover, more direct availability of nutrients for insects when we have the large herbivores.
Yeah.
So
You could just do.
Yeah, it's a basic question maybe as well. So in the second research, you had a nutritional quality of plants through time, and you collected that from ovarian specimens essentially, or like stored grass.
yeah.
Is there any relationship between time and like change in nutrient quality once you've harvested the plant, or is it just set once you've once you collected the plant? Because I work with DNA and obviously that degrades a lot.
Yeah.
But I don't know about those nutrients. Yeah.
Yeah. So the question is there a storage effect of on the plant tissues that are being saved over time, and could that affect the nutrient content? So we don't think so. So it's just this elemental nutrients that doesn't degrade over time. And yeah, I mean, there could be some very small amount of interaction with the gases, but there's been quite a few studies that have looked at these herbarium specimens on the just element amount, and people don't think it's changing it. But it would be ideal thing would be to every year do the analysis. Yeah. Maybe
in the first study about the fertilization, you were showing that if you added all nutrients, NPK and micronutrients, for large stimulation of the plant biomass or growth, but also if you just added phosphorus. But if you added phosphorus and potassium and micronutrients, you didn't get any effects. How do you explain that? Explanation.
Yeah, so the question is, if you had phosphorus in the meta-analysis study at the beginning, if you had phosphorus alone, you have an increase in plant biomass, but not when you add phosphorus, potassium, and micronutrients. So, of course, one thing could be a bias effect, that this is a meta-analysis and those are from different systems, different locations, and maybe they could be just due to those different dynamics in the different places. But, especially as we're going down into combinations like phosphorus, potassium, and micronutrients, unfortunately have a lower sample size on those studies, but it could be a real effect too. It could be that adding that additional elements even increases to a greater degree our nitrogen limitation in the plants, and then that in turn leads to no additional fertilization effect. Yeah.
Thank you for the talk. So there has been a lot of kind of discussion, and also there is evidence of nitrogen deposition, for instance, on the plant-neighbor interactions, lots coming from the US and in Europe. So these negative effect, right? So you have been looking into kind of interactions away from this device and then the grasshoppers and the plants as well. So in your study, in your sampling, have you actually gone just looking forward in looking into these interactions also with other insect pollinators? So do you have like linguistic what is going on also with other insects in these locations? Have you thought about it?
So looking at nitrogen effects on additional insect groups besides grasshoppers. Yeah, I don't know. For me personally, I might be stretching myself a bit, but I would definitely support that in a student project or anyone that wants to come to the site. I can help you find housing and all this. Yeah.
So if you've got a trend in sodium, how long has that been going on? Because it presumably it wouldn't be a salt panel or totally leached unless something's changed. So have you got a feeling for when something might have changed? Or is it mainly the CO2 you're ascribing the variations?
What's the response you're asking about?
So the level of sodium in the system, is there a long-term trend in availability of sodium, or is it mainly the balance of sodium to carbon that you're looking at?
So the trend that we looked at in the study is just sodium content of the plant, the grass tissue over time. So it's just direct.
That's been going on because it would have gone to zero or infinity if we've been for years. So there's something regulating it, presumably.
Yeah. So yeah, what's driving sodium deposition or sodium content in plants? Yeah, we don't know. I have supposed to be writing a paper on this topic or related changes in plant chemistry over time. The site has this national atmospheric deposition program in the US that there's a dry deposition measurements, and we didn't find any relationship between dry deposition of sodium and sodium content in the grasses over time. If one thing could be runoff from roads, but that's in the interior of the because it's not really near any roads that are being salted. So yeah, big answer is we definitely don't know. Yeah. Changes in when precipitation occurs, perhaps. Yeah.
Yeah, that's no question. I'll keep you on the channel.
Yeah.
But yeah, so I'm actually not sure like when we're supposed to end.
I think if we're done, yeah. You can ask me more questions. I'll be here. Yeah. Yeah. Yeah.
Great. Yeah, hopefully someone will stay around.
To remember to take my camera.
Yeah.
Yeah.
Okay, but you were also yes. I'm staying so can you fight the resist the temptations of going by the house? Resisted it.
You get a drink, but
Yeah. I do read
She couldn't well, Carly's somewhere else, but she was online all the time. Yeah, she was online. Yeah, that's. Okay, I couldn't see. I thought I was see here. I mean, well, that was it, Colin. No, I thought I was see by here. Yeah, okay. Yeah, no, I'm not sure where she is. She might be in the building somewhere. Yeah. I don't know. I don't know. Yeah. So when that's great. Fortunately, also last time it was great. Very nice. And I was
Yeah, you're gonna have to find a longer term.
But you can see that
No, I mean, the exchange rate is one. I know that's not true. I know.